Uday. I'm a professor in the biology department and graduate program director. And I'm excited to have this opportunity to introduce you to our current students so that you get a little bit of a sense of the research that they're doing. These students are found both in Winston Hall on the Ronaldo campus and at Wake downtown, so you'll see them in their natural habitats of those two locations, telling you a little bit about their work. Thank you and enjoy the tour of research interests.
Hi everyone, I'm Madeline Ward. I'm a first-year master's student here at Wake, but I've been here for a while. I did my undergraduate degree here. I'm currently working in Dr. Miles Silman's lab on the Project Surf, which is his core reef ecology portion of the lab. You can see on my monitor one of the beautiful pictures from our study site down in Belize. And so I focus most of my efforts on core reef ecology. More specifically, my personal research is on dietary partitioning of herbivores down at the reef. And in order to study that, I do a lot of work with camera trapping. I have recently been working on creating machine learning models to automatically identify the images that we capture on these cameras. And I'm starting some work also on DNA metabarcoding to study what fish are eating. In addition to studying coral reefs, a huge part of what this lab does is studying multiple aspects of ecology down in the Peruvian Andes. The two PhD students in this lab, Ellen and Cassie, both work down in the Peruvian Andes, and that's where the primary research that my advisor, Dr. Miles Silman, also does. He does a whole slew of research, including some really interesting work with paleoecology, work with illegal gold mining and the ecological effect of that. As you'll see in our lab, they also do a lot of work with drones and technology and remote sensing down in the Peruvian Andes. One of my favorite aspects of working in this lab is the community of our lab. We involve a lot of undergraduates in our lab work. That's how I got started with research. I started as an undergrad here at Wake in Dr. Michael Anderson's lab. I'm working with savanna ecology, in which I was super fortunate to be able to actually go into the field and do field work in Tanzania. And so I really enjoy working with undergrads. They've been a huge help in helping me identify fish.
Hi, I'm Jacob Cleary. I'm a second-year PhD student in the Molecular and Cellular Biosciences program, and I recently joined the Molecular Genetics and Genomics PhD program. I'm in the Keysang Reef Lab, and I study how methyltransferases and demethylases cross-regulate and interact with one another to control chromatin states. And broadly speaking, what that means is these proteins that are involved in chromatin, which is the functional unit of DNA, have tails, and these tails can be post-translationally modified, and those modifications dictate whether the chromatin is loose or tightly compacted. And loosely compacted chromatin is available for transcriptional machinery, which means you can produce mRNA transcripts, and tightly compacted chromatin means that it's inaccessible or primarily inaccessible to transcriptional machinery and is likely not transcribed. And I'm interested to see how these important proteins, the methyltransferases and demethylases, interact with one another or interact with each other's complexes to regulate the other's activity.
Hi, I'm Heather. I'm a third-year PhD candidate in the Cordy Lab. Our lab researches malaria pathogenesis and host pathogen interactions. We're really interested in looking at the metabolic changes during malaria and how that affects red blood cells and the parasite. We are a biosafety level 2 lab where we research Plasmodium falciparum. Our lab is a BSL2 or biosafety level 2 lab, so we follow all bloodborne pathogen precautions. In this lab, we have PCR station. We have a couple microscopy stations, one that allows us to do fluorescent microscopy. We have IHC station with a microtome. Then we have a tissue culture room with 2 biosafety cabinets where we do all of our blood and parasite manipulations.
Well, my name is Melina. I'm a second-year graduate student in the Moudet Lab at Wake Forest University. In the Moudet Lab, we actually work with a plant species called Arabidopsis thaliana. This plant just has a quick life cycle, which makes it a little bit convenient for scientists to work with. It also has the entire genome sequenced. That basically means that we know all the building blocks, or most of the building blocks, that go into making this plant what it is. So we actually work with a bunch of different mutant plants. This basically means that the genome or the DNA of the plant has been modified in some way. We actually utilize a bacterium called Agrobacterium tumefaciens, and in nature this bacterium actually causes infections in the plant, and it does so by inserting a piece of its DNA into the plant's DNA. So we actually take advantage of this ability, and that's how we basically make edits into the genes that we want, and it disrupts the function. And so we have these different mutant plants, and we're able to look at them under the microscope and basically see if they have some observable characteristic that might be different than the normal plant. And this is called phenotyping. So we do a lot of phenotyping in the Day Lab, which is why I'm on a microscope. And I'm currently studying a plant hormone called ethylene. So if you didn't know, just like us, plants also have hormones that are involved in regulating different processes from when they're seeds all the way until the end of their lives. So this hormone that I'm looking at is called ethylene, and it's a 2-carbon gaseous compound. It's actually involved in a number of different processes, but some of which you're probably familiar with, specifically fruit ripening and aging and flowering and stuff like that. So I'm actually looking at ethylene in the context of plant roots. So when we treat plants with the hormone, we actually use a precursor to the hormone called ACC because it's a little bit easier for us since it's not a gas. So when we treat with this precursor molecule, we actually get an induction in root hairs. So you can see that here. This is a control plant right here. So what you're looking at is this, I guess, longish structure. That's going to be the primary root. And then these smaller structures here, those are single cell projections called root hairs. So this is the control plant right here, right? This has no hormone added to it. And then when we do add ACC, you can see that there's a bit of an induction here, especially at the very tip of the root. And so that's currently what I'm focused on.
Hi, my name is Delcir Gamalila, a PhD candidate here at the Department of Biology, Wake Forest University. I'm in Michael Anderson's lab. So my current research focuses on understanding the patterns of seed production, seed predation, across the environmental gradients in the savannas. I also conduct experiments. As you can see, I'm in the greenhouse now, so I conduct these experiments in a controlled environment. The reason is to understand how variability in resources such as sunlight, soil moisture, affect the morphology, architecture, and survival or overall growth of these seedlings. And this is very important because it's very hard to control some of the environments to eliminate the disturbances such as fire, or herbivores, and other abiotic factors in the field. What I'm trying to do here is to mimic or manipulate some of the environmental variability that we observe in the field. So here you have what you can see is the treatment for shade, mimicking the effects of shade that these seedlings can get in the field. But also I'm treating some of them with a full light, and they have these color-coded telling me the amount of the treatment, about the moisture level, and the kind of species. So one of the species that I work with is Acacia totalis, Acatol, and Acacia robusta. So when I see the red one, I know this is for drought totalis, and this is a drought robusta. So this is my current research, but as I told you, most of the work takes place in the field. I use a range of data. Some of them are satellite acquired, such as the amount of rainfall that our experimental plots are in the field. But we bring some of the things here so we can understand the variability in resources in a controlled environment.
Hi everyone, my name is Mackenzie Brown and I'm a PhD candidate in Dr. Sarah Essman's lab. The SMIN lab is widely focused on protein-protein interactions that allow for rotavirus replication. Rotavirus is a double-stranded RNA virus, and as we know from the current pandemic, RNA viruses, such as SARS-CoV-2, has the ability to cause extreme health issues and economic defects. Specifically, rotavirus and other RNA viruses contain an RNA-dependent RNA polymerase, which is the polymerase that catalyzes important stages of RNA synthesis, such as replication. Our lab focuses on the regulatory partner of the polymerase, the core shell, and tries to use rotavirus as a model system to understand this RDRP regulation that occurs in multiple RNA viruses. Specifically, my project utilizes biochemical and structures of the rotavirus virion to inform mutants, BP2 proteins, the core shell protein, to then further understand how these mutants and their contacts regulate the activity of the polymerase and essentially rotavirus replication.